welcome back to plant developmental biology course. So, we are continuing root development and in today's class we will discuss vascular tissue development. So, basically vascular tissue is very important and it makes basically transport system in the plant, and transport system is absolutely essential for survival of a plant. We also have transport system in every organism. In animals, the nature of transport system also called circulatory system. It is because there is a circulatory mode of transport where we have a central organ called heart, but in case of plants, transport system is not circulatory, it is non-circulatory, and it is parallel. Two parallel tissues, xylem and phloems, basically makes the transport system. And xylem, what it does, it takes mostly water, minerals from the soil and it transport everywhere, and phloem basically takes photosynthetic material and lot of signaling molecules and it distributes or allocate to other part of the plants. Another important and key difference between the transport in the transport system between plants and animals are, in case of animals, transport occurs through the tube, which is either artery or veins. So, basically there is a very specialized structure which is called tube, and the transport occurs outside the cells, but in case of plants basically the transport occurs through the cells. So, there are spatial cells, they have acquired spatial features and the transport occurs from one cell to another cells, and it is moving for both in case of xylem as well in the phloem. And basically the vascular tissues have three different components: xylem, phloem and cambium or procambium. Procambiums are basically a kind of stem cell for generating other secondary tissues during the process of secondary growth, which makes cambium. But another important thing, that the organization of these tissues, xylem and phloem, is dependent on the tissue. So, if you look the root, there is a different pattern of vascular tissue arrangement. If you look shoot, there is a different pattern of tissue arrangement, leaf and everywhere. And the process or the developmental program for vascular development is set at the stage of embryogenesis itself, the primary vascular tissues. So, as you can see here, maybe in next slide you will see more detail, but these tissues are basically your procambium tissues, and these procambium tissues are going to basically make a kind of cambium cells. This is a root tip or primary root tip, and if you look the arrangement of this vascular tissue, so if you take from the outer side, you have epidermis, then you have cortex, and then below cortex you have endodermis, and after endodermis you have pericycle. So, basically your vascular tissue, they have pericycles, then in pericycles you have, this is a typical primary root of Arabidopsis, model plant Arabidopsis. You have xylem, and xylem has a kind of linear axis, you can see here, and in xylem you have protoxylem and metaxylem. So, these two are the protoxylem, they are the metaxylem, and then at the two poles you have phloem tissues. So, these are the phloem tissues pole, and in this phloem tissue there are two very important phloem tissue, these are called sieve element and then companion cells. These are very important. So, sieve elements and companion cells. And if you cut cross section at different places, the arrangement or organization of these tissues is different. So, the question is that how these vascular tissues are specified, how procambiums or vascular procambiums are specified. So, if you look the embryogenesis, early embryogenesis, so basically these are your ground tissue initials are the putative ground tissue they are going to take, and then these are the vascular stem cell initials. And what happens here at this stage, this is maybe more clear. So, these are ground tissue precursors, and then you have pericycle, and then you have vascular stem cells. And again I would say that auxin is very important in all this kind of cell specification and patterning, and auxin is basically polar transported by pin proteins. So, these pin proteins are very important in distributing auxin. Basically helping in a particular tissue or in a particular organ to have auxin maxima. And another important thing for these spin proteins are that these spin proteins are asymmetrically localized in the cell. So, in one cell wall they are localized, and their asymmetric localization basically provides a direction for polar auxin transport, because they are the transport proteins. And if you look at the early stage, what happens that auxin is polar transported, and this polar transport activates a class of transcription factor. This is MP, is again a kind of auxin response factor, and this MP activates a key regulator of procambium identity or specification, which is HB8. HB8 is again homeobox class of transcription factor. So, this is how the program of procambium specification takes place at embryogenic stages. In later stages, in the root, if you look how the procambium state or stem cell fate is maintained or regulated. So, this is a cross section, a typical cross section of root. So, this is your xylem axis, this is the phloem pole, and these are the procambium tissues. And what happens that there is a signaling between phloem cell and procambium cells. So, from phloem cells there are two prototype signaling molecule 41 and 44. They are getting received by the procambium cells through PXY and TDR based reception mechanism, and this eventually activate another class of homeobox protein which is called VOX4, and VOX4 basically helps in specifying stem cell fate to the procambium. Then how tissue patterning is happening. So, auxin and another very important hormone is cytokinin. Their antagonistic interaction ensures a proper tissue arrangement in the root. So, if you look how auxin and cytokinins are distributed in the root, these are IAA2 promoter driving GUS. IAA2 gene is a target of auxin signaling pathway. So, which means that wherever this expression is there, it means that those cells have activated auxin signaling program. And this is ARR5, which is response regulators, and response regulators are basically downstream genes regulated by cytokinin signaling pathway. So, if you see expression pattern here, you tells that these are the cells where cytokinin signalings are active. So, if you look here in the primary root, what happens that auxin is very high in the xylem axis with little in the procambium cells, and in xylem axis protoxylem are having slightly more than the metaxylem, it looks like. However, if you see the cytokinin signaling are very less in the xylem tissues, but in procambium tissues the cytokinins expressions are high. And this kind of pattern, this is a kind of antagonistic pattern, basically defines a regulatory mechanism for different tissues. So, what happens in the protoxylem cells, what happens, you have a very high amount of auxin, and auxin basically activate AHP6 protein, and this AHP6 protein, it goes and inhibit cytokinin signaling in the protoxylem. So, in protoxylem, for protoxylem to be as a protoxylem, what has to happen, there should be very high auxin signaling and no cytokinin signaling or very low cytokinin signaling. Another thing what happens that this auxin activates TMO5 and LHW, and this activates LOG4. LOG basically a gene which play a role in cytokinin biosynthesis and which results in cytokinin biosynthesis. But this happens in the protoxylem, but this cytokinin move to the procambium, and in procambium the cytokinin signaling is getting activated, and this cytokinin signaling in the procambium cells activate all the genes or all the programs which is required for procambium cell maintenance, whereas this auxin signaling is working in the xylem axis and regulating the differentiation of xylem tissues. Another regulatory mechanism which works in defining cell fate or identity of xylem cells is through short root and scarecrow protein, and this is very important. So, if you look here and if you recall your previous class, what happens that this short root protein is synthesized in the vascular tissues. So, you have vascular tissues, these are the vascular tissues. In vascular tissues you have synthesis of short root protein, but short root protein then moves to the endodermis. So, this is your endodermis, and in endodermis where short root protein moves, so protein is made in the vascular tissues and it is moving to the endodermis, and in endodermis it get nuclear localized, and once the short root protein enters inside the nucleus, it activates scarecrow, and scarecrow and short root protein they together activate microRNA 165, 166, and these microRNA they are basically getting produced in endodermis and then they are diffusing back in the vascular tissue, and this diffusion basically what does, it creates a kind of gradient. So, if you look here, so the cells which are directly are the cells which are closer to the endodermis will receive high amount of microRNA, and the cells which are away from the endodermis they will receive low amount of microRNA, and that is why you have a gradient of microRNA higher to lower side, and this gradient of microRNA is also reflected in the targets of microRNA. So, if you look here, what happens, this is microRNA. So, microRNA level is very high in endodermis and then microRNA level is going down once you move through pericycle, protoxylem and in metaxylem. So, in metaxylem what you are expecting, very low amount of microRNA, but in protoxylem you are having high amount of microRNA. But if you look the target, so basically this microRNA targets homeodomain containing transcription factor, fabulosa, and that kind of transcription factor we have seen in one of the previous class. But what happens that the metaxylem will have high amount of homeodomain proteins and then the protein levels will go down. So, the cells like pericycle or protoxylem, let us see between protoxylem and metaxylem. So, protoxylem is going to have high amount of microRNA, but low amount of HDG protein. Metaxylem is going to have low amount of microRNA and high amount of HDG protein, and the amount of HDG protein basically defines the identity of a cell. If HDG protein is low in amount, the cell is going to be protoxylem, and if HDG protein is high in the cells, it is going to be metaxylem. So, basically these kind of regulatory mechanisms is defining xylem cell identity. So, apart from these specification and identity, there are another very special feature which is associated with the xylem cells, which is a kind of secondary wall formation. So, if you look normally what happens that if once your cell is specified as a xylem cells, this xylem cells enters in the process of secondary wall patterning, and there are lot of changes occurs, and eventually the xylem cells they die through the process of programmed cell death, and this is very important because xylem has to work as a tube for conducting water and other minerals, or it has to have a very strong mechanical property to be able to transport this. So, this process of xylem differentiation is also regulated by another genes. Some of these genes are VND6 and 7, SND1, and these are LOB domain containing transcription factor, and they are eventually regulating some MIB domain transcription factor, and this interaction of all these proteins basically they ensures that there are some cellular modification by depositing some cellulose, xylon, lignins, and then eventually some of them are regulating the final process of programmed cell death to generate a final functional xylem tissues. Now, another important tissue which is part of vascular tissues are phloem tissues. If you look here, so this is the picture which is which tells that they are running in the parallel. So, this is basically your xylem vessels and this is your phloem tissues. In phloem tissues there are two important tissues, one is called phloem sieve tubes or sieve element or sieve cells, and this is directly connected with a companion cell from one side and phloem pole pericycle cell from another side. So, if you make a cross section here, you can see that these are the sieve elements, these are the companion cell, and these are the pericycle cells which is directly attached with the phloem pole pericycle cells it is called. And early stage of this phloem development is regulated by this mechanism. This has been identified. OPS CLE40 mediated BAM regulation, which this is also working in the dose dependent manner, and BRX based mechanism. So, these mechanisms are these regulators together they are functioning to ensure a proper proto-phloem specification. Proto-phloem specification, these phloem at very early stage in the meristematic zone or up to elongation zones are basically called proto-phloems. These proto-phloems then undergo the process of special differentiation program, and this differentiation program is highly regulated again, which we will see later. But another very important regulator which regulates phloem development is another transcription factor which is called APL, Apple transcription factor. As you can see that, and this regulation of phloem is very important. If you do not have phloem, so in this Apple mutant phloem identity is totally lost, and this mutant is seedling lethal. So, plants are grown, but it cannot survive for the entire life. And if you look what kind of phloems are defective in this mutant background, so these are the markers. J0701 basically marks your sieve element or proto sieve elements, and if you look in normal wild type root, you see these markers are present, but in Apple mutant these markers are absent, which tells that the sieve elements or proto sieve elements are missing, or their identity is not established. The SUC2 protein, and SUC2 is a marker for companion cell, and you can look here that even companion cells are not formed in the Apple mutants. So, this tells that Apple regulates both the cell types, sieve element as well as companion cell identity in the root. And if you make a cross section, this is your wild type, you have xylem axis, then you have this phloem cells. But if you look the mutant background, this identity of phloem cells are totally different, but if you complement with the Apple, you can see again the normal function. Then you know that Apple is regulating both companion cell as well as sieve element, but where does Apple itself express? So, there are two way of regulation. One regulation is that which is called cell autonomous regulation or non-cell autonomous regulation. So, basically what happens that if the regulator is present in the cell and then regulating its identity, then it is called cell autonomous. Protein is present in one cell and regulating identity of the neighboring cell or some different cell, then it is called non-cell autonomous regulation. And when you check the expression pattern of Apple protein, it expresses both in proto-sieve elements. This is basically nuclear localized signal. You can see in the sieve element, this is proto-sieve elements, and later stage you can see that this is your proto-sieve element, and then the companion cell in the differentiation zone, these are the companion cell. So, this express both in sieve element as well as companion cells and regulates their identity. Then next, okay, so this is a regulator of both the cells. Do we have some specific regulation of the sieve element cell? To identify this, there was a genomics based approaches taken. So, what it has been done? So, there was approach called microarray. If you recall, we have discussed in some of the previous class, and what happens that microarray has been performed, but it was performed in a very specialized manner. These phloem cells were sorted using a technique called FACS. FACS is fluorescent activated cell sorting based techniques. So, if you put some fluorescent tag in these cells, then specifically you can isolate these cells from these tissues. And then phloem cells are the cells which are present at the place of phloem has been isolated from Apple mutant, and then total RNA was extracted and it was used for doing microarray and to identify what are the genes whose expressions are getting affected. And out of these genes, so there were large number of genes, but two important transcription factors were identified. So, one was NAC45 and another was NAC86. So, both are MAC domain containing transcription factor NAC45 and NAC86. And okay, so basically what happens here, as you can see that in Apple mutant, basically this is expression of NAC45, but NAC45 expression totally absent in the Apple mutant. Similarly, if you look here, this is APL mutant and NAC sensitive, so cells are absent. So, this tells that the both NAC45 and NAC86 are down regulated in APL mutant background. Then if you check the expression of NAC45 and NAC86 itself, what you find that these genes are very specifically expressed in the sieve element, but they do not express in the companion cell. So, this tells that they might be they might regulating sieve element cells, not the companion cells, and this was true when you make mutant single mutant of NAC45 or NAC86 did not give any phenotype, but when you made double mutant, what you can see that there was a defect here. And then if you analyze these mutants or the wild type pattern of sieve element differentiation in details, what happens that if you look, so this is basically tracking one single layer of sieve elements, and what you can see, this is towards the root tip, this is towards the upper side, and at the root tip sides you can see that sieve element cells, they are looking very normal in the meristematic zone, and then suddenly what happens that there is a cell elongation, the cells started elongating, and then some changes started in the phloem cells, and these changes immediately turns in a way that this cell degrades all the cytoplasm including the nucleus. So, sieve element, the functional sieve element is anucleated, there is no nucleus, most of the cytoplasmic components are degraded, and this is essential for its function of transport. As you know that transport is occurring inside the cell. So, cell has to have minimum inhibitory components here for the transport system, and this happens in a very specific manner. How this is? So, if you look, there are three stages. First stage, it is very normal. Second stage, which is called intermediate, the process of differentiation or special differentiation of cell sieve element started, and then in stage 3 the differentiation has occurred in a way that the sieve elements has totally lost the nucleus. Now it is empty, more kind of channels, and the common cell wall between two cells are very porous in nature in case of sieve elements. That is why it is called sieve, because there are holes, there are pores in the cell wall, and it makes a kind of a structure like sieve, and that is important for water or anything to move from one cell to another cell. If you look the mutant, this double mutant, what we see that cytoplasm and nucleus degradation is blocked. So, there is no degradation. Since there is no removal of nucleus, there is no removal of cytoplasm, the sieve elements are not completed its differentiation program, and that is why these plants are also seedling lethal, they do not survive for lifelong. And you can check here, so this is a longitudinal view of your root tip. You can clearly and very specifically see the sieve elements of, because of its a very special feature. You can see that it has a very thick cell wall which distinguishes from other cells. And if you look, this is Cal7 promoter, which is specific for the sieve element, and if you drive, H2B is histone 2 protein. So, basically this is a nuclear localized protein. You can clearly see that till here in the sieve element profile there is nucleus, but here onwards this is the sieve element cells without nucleus. So, nucleus is totally disappeared. This is the enlarged view. So, if you look here, up to here there is a nucleus, but here you can see there is a diffused GFP signals, means nucleus is under the process of degradation, and then the just next cell here you can clearly see that nucleus is almost removed. But if you look the mutant, in mutant what happens that nucleus is not degraded. Even if you go to the very higher side of the cells where the sieve element cells are very long, but you can still see the nucleus. This suggests that in the double mutants nucleus is not degraded. And then there is a mechanism. What is the mechanism exactly which is regulating the process of nucleus degradation? This was again to identify. So, NAC45 and NAC86 is regulating nucleus degradation, but it is a transcription factor. It cannot go and directly regulate the process of nucleus degradation. It must be regulating some of the genes which is directly involved in the process of nuclear degradation. To identify these genes, another round of microarrays was performed using this double mutant and NAC45 overexpression. And from this microarray analysis, there were 4 genes, very small family of putative nuclease family of genes were identified, which was NEN1, NEN2, NEN4, 3 we are showing here. And what you can see, there is a very important thing what is happening. First thing that these genes are expressed in the sieve element, which is basically correlating with the fact. And another important thing, if you look the sieve element at early sieve elements or young sieve elements, the gene is expressed in the sieve element, protein is made, but protein is not able to enter inside the nucleus. This is the nucleus, and protein is entering only in the cell where nucleus has to be degraded. So, before these cells, protein is not able to enter inside the nucleus. So, there are two regulation. One regulation is that these nucleus genes are getting expressed in the sieve element, that is regulated at the transcription level, and then another regulation is at the post translational level. When protein is already made, its translocation to the nucleus is regulated. Similar kind of things you can see for NEN2 as well, whereas if you see NEN4, this is specifically expressed only in the cell where nucleus has to be degraded. So, if you look, the early cells does not express, later cells nucleus is degraded. So, there is no nucleus, no signal, but this cells has NEN4 protein localization only inside the nucleus. So, all these stories tells that these putative nucleases might be responsible for nucleus degradation, and this is supported genetically. When you have NEN4 mutant background, you can see that nucleus degradation is defective in this mutant background. So, here everything is normal except NEN4 is not present. So, if you take all these together, we suggest that apple is regulating both sieve element as well as companion cell, and then during this process apple is regulating NAC45 and 86. NAC45 is regulating sieve element differentiation, but cytoplasm as well as nuclear degradation, but it is regulating NEN1 to 4, and this NEN is specifically regulating nucleus degradation. So, this is how the process of phloem specification takes place to make a mature mature sieve element cells. And as I said, this mature sieve element cells is very special. It has this sieve pore like structure. This is almost empty. So, it is very suitable for very high or efficient transport system. So, in general, to summarize the vascular development, so what we have studied, there is a auxin. Auxin is regulating auxin response factor MP, and it is regulating ATHBH4, and this process is basically ensuring vascular stem cells through the VOX4 genes. There is another regulation from the phloem sites, which is CLE1, CLE41 and CLE44 mediated regulation, and it is working through PXY TDR to regulate the stem cell identity. And then later on, what is happening, there is a mechanism through short root protein and scarecrow protein, microRNA and HDG mediated protein. This is basically regulating xylem cell identity, protoxylem versus metaxylem identity. Another regulators, which is VND7, which is regulating again protoxylem identity, VND6 regulating metaxylem identity. But on the other hand, if you look apple genes, apple is basically activating phloem kind of identity, apple is regulating companion cell and sieve element both, and for sieve element based specification it is using NAC45, NAC86 and NEN mediated signaling pathway. And then protoxylem is also regulated by the feedback of auxin, AHP6 and cytokinin based mechanism. So, we will stop here. In next class we will discuss root branching. Thank you very much.